So it's already 15 past 10. So let's go ahead and begin our discussion where we left the last time. So I'll be sharing my screen and you will let me know whether it is visible to you all or not. Okay, so is my screen visible?
Yes.
Okay, good. So what I will be doing is today we will be talking more about where we left the last time. We we have to discuss now more about the technical details of the transcription reaction and how it takes place and what is the guiding force, or you can say which is the enzyme that is involved in the process. That's what we are going to discuss today. So till now we have talked something about the necessity of a messenger molecule or a transient molecule that is to be synthesized before before protein synthesis, that is. So and also we talked about the hypothesis which actually was given by Crick, and then the hypothesis was tested and found not worthwhile, and hence there was an alternative hypothesis that was given by the group of Jacob, and they proved that there is existence of a transient molecule which they referred as the messenger RNA later. So from that point onwards, we will move ahead. This is just a general, what you can say, way of looking at transcription. You can divide the entire transcription reaction into three phases. First is the initiation phase. Next is the elongation phase, and the last is the termination phase. So initiation phase can be broadly divided into two sub-stages. One, when the RNA polymerase, which is the enzyme that is needed for transcription, recognizes the region from where it is going to begin the process of transcription, and that region is referred to as the promoter sequence. And further, once it has recognized the promoter, then it follows the next step of the synthesis of RNA molecule, that is starts it by the synthesis of first phosphodiester linkage, the NTPs, which are ribonucleotides, which are used for synthesis of new RNA molecule, utilizing the DNA as the template. Next, once this initiation steps are done, then the RNA polymerase progresses into the elongation phase, where it is essentially just incorporation of new nucleotides, taking the DNA as the guide, to synthesize a complementary RNA molecule. And finally, when the process reaches to a site which is referred to as the terminator site or the termination site, it is again a sequence which is very similar to that of promoter. That means like promoter, it's not exactly the similar sequence, but like promoter, it is also a sequence of DNA nucleotides, which is referred to as the termination site, where RNA polymerase leaves the DNA as well as releases the newly synthesized messenger RNA molecule. So, is there any doubt in this entire process? That this is the overview of the transcription reaction. So, is there any Any doubt in this basic process of transcription? Or are we good to go ahead from this point? Yes? Am I audible to all?
Yes, sir.
Okay, good. So the process of transcription can be summarized into three phases: initiation, elongation, and termination. That's what you need to remember to understand the process, what we are going to talk about in the next slide. So as we have seen in the previous slide itself, that basically the synthesis of new RNA molecule is catalyzed by an enzyme that is called as RNA polymerase, and hence it became a very important what you can say scientific requirement that one should know something about what is RNA polymerase, how does it look like, and what are parts or subunits of RNA polymerase exist. So obviously, the first steps were taken in order to isolate the E. coli RNA polymerase. This RNAP is just a short form of RNA polymerase. Okay, so E. coli RNA polymerase was found to contain, uh, that means functional RNA polymerase was found to contain basically four subunits, two which were very similar in size. They were named as beta and beta prime subunit. They were having almost the same size. So one was, that is beta was 150 kilodaltons, while beta prime was 160 kilodaltons, respectively. Apart from this, RNA polymerase was also found to contain two more important subunits. One was called as the sigma subunit or the sigma factor, and the alpha subunit. Now, all in all, when they found out that what is the stoichiometry is in what way they are arranged together, then it appears that it consists of two alpha subunit, one beta prime, one sigma subunit. This is the composition of RNA polymerase. That is the prokaryotic RNA polymerase. In some texts, you will also find that it is mentioned that it also consists of an omega subunit along with this, but that has a very little, what you can say, importance in terms of the activity of RNA polymerase, and hence it is all recommended that we should refer to RNA polymerase as consisting of these many subunits only, which are given in the top. That is two copies of alpha subunit. So it is written alpha 2, one beta, another beta prime, which is similar to beta subunit, and one more, that is the sigma subunit. Okay, so this was first done. That means isolation of all the subunits in order to understand what is the subunit composition of RNA polymerase or stoichiometry of RNA polymerase. It was done by the two gentlemen, Travers and Burgess. This was done in 1969. Travers and Burgess together actually isolated all the RNA polymerase, E. coli RNA polymerase subunit using a very simple electrophoretic procedure that is called as SDS page. Okay. Now, when we know that it is this composition, then it was found out that the basic activity of RNA polymerase is still retained, although it can give away once it gives away its sigma subunit. So it was said that the basic RNA polymerase activity composition is alpha 2, beta, and beta prime. So this composition is referred to as the core enzyme. While when this is coupled with, that means when the core is with sigma subunit, then this is referred to as holo enzyme. Now, holo means complete. Okay, so the complete enzyme is the core, that is alpha 2 copies, beta and beta prime, along with sigma subunit. While the basic RNA polymerase activity is still retained, although you remove the sigma subunit from the holo enzyme. So what remains is basically referred to as the core enzyme. So is this fine, the nomenclature? Or should I repeat it again for you all? Can anybody tell me what is the difference between the core enzyme and the holo enzyme? Yes? Anyone?
Sir, core enzyme is without the sigma unit with
Yes.
alpha 2 and beta prime, and holo enzyme is with the sigma unit.
Okay. So all in all, why it is named like this is because this is the minimum composition of RNA polymerase that can still give you the activity which is associated with RNA polymerase. So this is referred to as the core enzyme. However, when we add the sigma subunit to the core, then it is referred to as the holo enzyme. Holo means it's a complete enzyme subunit composition. Okay. So from this point onwards, we will move ahead. Now, just pay some attention here. When it was found out that when you compare the transcription activity of the core enzyme, just look at this. Okay, when you compare the transcription activity of the core enzyme with the holo enzyme, then you can find out that when you give it a native DNA molecule, native is the intact DNA molecule. That means there are no breakages in the DNA. It is what you can say, unbroken and well-to-do DNA molecule. So it was taken from the T4 E. coli phage. Okay. Now, when you give native DNA molecule, intact DNA molecule to the core enzyme and to the holo enzyme, which is seemingly giving you better activity. Yes. If you look at the values given over here, this is the activity of the core enzyme. This is the activity of the holo enzyme. Which of the two seems to give you better activity when you are giving both the enzymes, the core enzyme as well as holo enzyme, intact native DNA molecule? Did you get the question?
Sir, holo enzyme.
Holo enzyme seems to give you better activity, right? So now they tried something else. What they tried was that along with sometimes it was found out that if you replace the intact DNA with a nicked DNA is something which is, say, imagine that this is a double-stranded DNA molecule. Nicked DNA has some breaks in between. These are single-strand breaks. These are not double-stranded breaks. Okay, these are single-stranded breaks. So you can imagine that the nicked DNA may look somewhat like this. Okay, so there are single strand breaks here, as you can see. Okay. Now, when it was compared to the intact DNA, when nicked DNA was given, it seems that the activity of the core enzyme increases, right? While activity of holoenzyme is more or less same, correct? Can you see that? That when you look at the level of activity of core enzyme with intact unbroken DNA, with the nicked DNA, it seems to have raised, right? There is more activity here, while there is less activity here in case of normal DNA compared to the broken DNA, right?
Yes, sir.
Okay, so it was reasoned like this, and perhaps both can give you better activity if at all there is some kind of a DNA breakage. While holoenzyme can give you the same activity no matter whether it is broken or whether it is unbroken DNA, and you are providing it to the holoenzyme. Putting it other way around, we can say that the holoenzyme is able to transcribe intact DNA better compared to core enzyme. Do you agree with me?
Yes, sir.
Okay. Now we move ahead from this point. Okay. So now we have established this that the RNA polymerase, what is the composition of RNA polymerase? And we also found out that how you have two different forms of the active enzyme. One is the core enzyme and another is the holoenzyme. And when we compare the activity of the two forms of the enzyme, then it seems that core enzyme is deficient in transcription of intact DNA molecule, while it can transcribe the DNA only if there are some single-stranded nicks in the DNA. The DNA is broken. Other way around, holoenzyme can transcribe intact DNA in a much efficient manner. Okay, so considering this as our fundamental understanding about the activity of the core enzyme and the holoenzyme, let us find out that why is it so that the core enzyme is transcribing lesser compared to the holoenzyme. Okay, so to do that, what was done was that there was a study that was initiated by Watts and colleagues. Okay, now a simple question was asked first. So the difference between core enzyme and holoenzyme is only of the sigma factor, right? Holoenzyme contains the sigma factor while core enzyme lacks the sigma factor. Correct? So, and because holoenzyme was capable of doing transcription with both the broken DNA as well as the unbroken DNA, it was supposed that, or it can be considered that sigma is responsible for providing the specificity of transcription, which is perhaps attributed only to the holoenzyme and not to the core enzyme. Okay? Are we good to go ahead from this point? Yes?
Yes, sir.
Okay, good. So now what Watts and colleagues did was that in order to show that perhaps sigma is providing the specificity for transcription, Watts and colleagues initiated a new study wherein they they used holoenzyme and they performed a transcription reaction of the T4 genes. Now, And on the basis of that, they concluded that perhaps 4 doesn't have the specificity of transcription which is necessary for RNA polymerase, and is only the specificity is only associated with the holo enzyme. So it is obvious that perhaps sigma factor is providing that specificity. So to do that, what they did, let us understand first. Okay. Now, how did they prove that sigma factor or the holo enzyme is more specific in doing transcription of certain genes of T4 DNA molecule, but will not do the transcription of all the genes? So first of all, you need to understand that T4 is a T-even phage. And what's the property of the T-even phages, if you remember? If you remember, bacteriophages can be broadly classified into 2 categories: lytic phages and lysogenic phages. So, in which of the 2 categories will you put the T4 phage? Yes, lytic or lysogenic? Okay. There are no responses coming from your side, so I will tell you it is a lytic phage. Okay. And when we are talking about the lytic cycle, then if you remember, then in case of a bacteriophage, what happens is that, consider that this is the bacterial cell. It sits on the top of the bacterial cell, right? Somewhat like this. Sits on the top of the bacterial cell surface, and then it injects the viral DNA molecule inside. So this is what is the T4 DNA. Once it gets inside, then there are certain phases in which the viral genes are transcribed, and there is a logic behind it. So that's what I'm going to explain you first, and then we will find out that how did Watts and colleagues actually establish this fact that only immediate early genes are transcribed by holo enzyme, while core enzyme doesn't have any such specificity. Okay. So whenever the viral DNA gets inside, then there are 3 types of viral genes that are transcribed. The first to get transcribed are referred to as immediate early genes. So the first ones are immediate early genes. Next in line, that means once the products of immediate early genes are available, then transcription of delayed early genes begin. Okay. So first are immediate early, next are delayed early. Okay. And when the products of both these genes are available, then the third ones are called as the late genes. These are transcribed the last. So if I group it under timelines of infection, then within the first 2 minutes of infection, those are immediate early genes which are getting transcribed and the products are being formed. Once the products of immediate early genes are available, then between 2 to 5 minutes, the delayed early genes are transcribed. And further beyond that, after about 10 minutes of infection, the late genes are transcribed. This is the sequence in which the transcription process actually occurs after the virus has infected the bacteria. Is it fine? Should we go ahead from this point? Yes?
Yes, sir.
Okay, good. So now once we know this, then the task is now to find out which genes are getting transcribed when I'm giving the holo enzyme intact T4 DNA. Now this is completely in vitro reaction. Okay. There is no such process that you are infecting E. coli and then you are looking at it. Okay. So this is a completely in vitro reaction where you give isolated T4 DNA only and you mix it with holoenzyme and give it all the raw materials which are needed for RNA synthesis. Is it fine? This is one reaction. While the other reaction composition will be T4 DNA, and instead of holoenzyme, there will be core enzyme. So either you add holo or you add the core enzyme. Okay. Is it fine? Okay. Now your job is to find out that whether it is the immediate early genes that are getting transcribed, delayed early genes that are getting transcribed, or late genes that are getting transcribed, right? So how will you find that out? Did you get the question? Think about the situation in a bit more detail and answer the question. Yes? Anyone?
Sir, can you repeat the question?
Okay, fine. So now your job is to find out, okay, just to find out that which of the two enzymes are better in terms of transcription specificity. Watts and colleagues have worked out this kind of reaction, in which what they are doing is they are giving native T4 DNA, that means intact T4 DNA, to holoenzyme as well as core enzyme, along with all the raw material that is needed for transcription. Okay. Now their job is to find out from the products of the reaction, which is coming from the core enzyme activity and the holoenzyme activity, they need to find out that which mRNAs are getting synthesized. Whether these are coming from immediate early genes, delayed early genes, or late gene products. Is the question clear?
Yes, sir.
Okay, so now you will tell me how they will identify the mRNAs, whether they are coming from immediate early genes or whether they are immediate early gene products or delayed early gene products or late gene products. That's your job to find out now. So how will you find out whether these RNA molecules are the products of immediate early genes or delayed early genes or late genes?
Sir, you are saying that there are two different conditions, one with holoenzyme and another with core enzyme.
Exactly. So there are two tubes that you're running. One tube consists of T4 DNA with holoenzyme and all the raw materials for transcription. And the other tube consists of T4 DNA with core enzyme and all the raw materials for transcription.
Sir, so the one with holoenzyme transcribes immediate early genes.
Yes, but you need to confirm that they are immediate early genes only. So the point is that, okay, we made the statement, that means what was obtained by Watts and colleagues. But now you need to put in yourself into the shoes of Watts and colleagues and try to find out that how they might have assessed, okay, I got mRNAs as the products. I will get mRNAs in both the tubes, right? Because even core enzyme is capable of doing some degree of transcription. Now, my job is to find out whether these mRNAs that I'm getting are the products of immediate early genes, delayed early genes, or late genes. So, how will you find it out? That's the question. Yes. So now your job is that I have given you the RNA products of both the tubes, and I am asking you that, okay, tell me that whether these RNA products are coming from immediate early genes or delayed early genes or late genes. So how will you do that? How will you find out that from which genes these RNAs have been synthesized? So that's your job to find out.
So the survey, the RNA will have somewhat similar genes to the immediate early genes.
Yes, exactly. So they will have the sequence which is similar to the sequence of immediate early genes or delayed early genes or late genes. So now your job will be to find out that which RNAs are similar to immediate early genes, which RNAs are similar to delayed early genes, and which RNAs are similar to late genes. How will you do that? You cannot do sequencing, right? It's pretty much late in the history that sequencing actually became a conventional technique. At the time of Watts and colleagues, this was somewhere in the era of 1965. At that time, there was uh, no route by the way of which you can actually go for DNA sequencing. DNA sequencing or RNA sequencing is not possible, not the choice. So what you will do? Actually, Neha has given you a very good insight in terms of thinking in the direction where you should think in order to find out that RNAs belong to which genes. Okay, can you get to the answer? Okay, what I will do is this. What if I leave you today with this question, and I'm going to post something in the Google Classroom. Just take a look at it, okay? And we will discuss about the answer. If you can figure out the answer, nothing like it. But if you cannot, we will talk about the answer in tomorrow's class. Does that sound good?
Okay, sir.
Okay, so I will be giving you a kind of a direction in which you need to think. Okay, by putting some kind of a material in Google Classroom, so you can refer to that material and think in the direction, so that you can find out that these RNA products are coming from which genes. Does that sound good?
Yes, sir.
Okay. So good enough. So what I will do is that I will end the class today here, and to look into what kind of the results you can expect and how you can figure out that which genes are getting transcribed. Watts and colleagues used a very interesting technique. So some lines to think in that direction I will put up in the Google Classroom, refer to the text there, and from that figure out that how you can find out that what kind of products mRNAs you are getting from holoenzyme activity and from coenzyme activity. So that is it for today. So in case you are having any doubts, you can ask me right away, or else I will put the material in the Google Classroom and then we can discuss the rest of the things.